Thank you for giving me the opportunity to talk about this project that we did when I was still at LifeArc. This is a project that is going to highlight some of the use cases of Nanome for antibody as well as peptide visualization. And hopefully you will be able to follow the project and see how the structural information was very useful for us to design a vaccine for Alzheimer's. And just to highlight how you could look through the NMR conformations in Nanome, I'm just going to show you how you'll be able to scroll through the alternative conformations of this peptide. I'm just going to play it like this so you can see, although it's not an MD simulation, it actually looks as though you're looking through the trajectory of an MD simulation. These are 20 conformations of the peptides that had been identified before we started the project. This peptide is believed to adopt many conformations, but most of the conformations that are known have got this alpha helical structure here in the middle. And the rest of the peptide kind of forms unstructured coils around. As you can see, we've got the peptide in its kind of resolved structure. So this structure was resolved in 2012. And people understood that somehow, although they did not know which conformation was responsible for causing amyloidosis, this beta amyloid peptide seems to adopt conformations that do form amyloidosis. And as you can see, there's this conserved amino acid just for us to be able to see which is the N terminus and how long the peptide is. We could actually use Nanome to visualize that. So I'm just going to display I'm just going to highlight this and then display for you so you can see. We will first label. Let's display the labels so we can see the amino acids with the residues labeled. Get the residues labeled, but we can also look at the actual residues themselves. As you can see, this is as we start from position 1 here, where the aspartate is, and then you've got the glutamate in position 3, and this is the beginning of our N-terminal of the peptide, and then it forms this kind of loop here, where glutamate 11 sits at the position where it turns into this alpha helical conformation. And this alpha helical conformation is preserved, as we saw in the kind of, when we're scrolling through the NMR conformations, that this is all preserved. And if you wanted to just have a look at that again, here you go, you can see that is always preserved as an alpha helical conformation. And these here kind of adopt different conformations, although there is some identification that there might be some favored conformations, but as you can see that the variation is not here, variation is out here and at the end. As you can see here, we've gone all the way up to position 40. Let's stop this so you can see where we've gone up to. This is the last resolved amino acid in this chain. This protein actually has 43 at maximum and 36 at minimum in most of the observed peptides that are known to be part of these beta amyloid peptides. So this is what we had, and this is the information that we had when we started at LifeArc.
Is there anything you want to say about how this paper came together?
Just to highlight some of the things from the paper which were interesting was, as I said before, we took this peptide and what we did was we cut it to position number 18. And then we generated the reason why we did this is because in literature, there had been other antibodies that had been generated against the full-length peptide. And those antibodies were not known to be effective in the disease model, although they bound to the beta peptide. So what we ended up doing was looking at subsection of the peptide in particular of interest to us was from position 1 to position 18. And we wanted to do this because there were no antibodies that had been generated for this particular part of the peptide. So we truncated it in position 16, and this part was used to immunize mice. And the mouse themselves produced an antibody that is referred to in this paper as TAPO1. And TAPO1 was seen to bind to this to the full length of the amino acid, of the peptide. And so we discovered an antibody that actually bound. And the next part of the story was interesting because although this antibody was binding here, we then decided, because we didn't understand why this antibody was actually binding with such a low affinity to this, we decided to try, and it was binding at about 90 nanomolar in terms of KD. And it was also very far. It was also binding, the binding profile for it, when you looked at the biocore binding, and this is SPR assay, you could see that although it was binding tightly quite quickly, it would just dissociate quite quickly, which is very, very surprising for a peptide. Normally, antibodies or peptides really bind to proteins with a very low off rate. So this was quite surprising for us. So we wanted to understand what the problem was with this particular antibody and why it was not showing the typical profile of high affinity and longer residence time of binding. So we were very lucky because Gareth and the team managed to generate the structure of the peptide bound to the antibody. Let me just present that structure to you guys. After my crystallographers managed to get the crystal structure, we looked at the structure that was available to us. And lo and behold, two most interesting things about this particular thing. Number one, we saw that the actual conformation that had been trapped by the peptide, as you can see here, is a novel conformation. If you compare this conformation with what we had seen in here, actually you don't have that cyclized part. And in fact, if you're comparing this peptide, we're starting here at position 3 or position 4 up to position 12. You can see here that we go all the way up to position 12, and this is linear. But in our peptide that we have found to be bound, this was in a kind of hairpin turn. And actually this was very interesting because this conformation had not been seen. And as I was showing you through the available NMR structures here, this actually is always conserved, this this alpha helical structure here. The two surprising things were effectively that we don't necessarily, with this kind of short peptides, you don't necessarily expect it to adopt something completely different, especially when it's not fully bound. Because when you see here the interaction between the antibody, you can see that the CDR regions, and we're going to talk about those in a minute, actually there is no lead that's catching the conformation in that position. Almost suggesting that this conformation is something that maybe is sampled in nature, but the antibody recognizes it and captures that conformation. So that was of interest to us, because if this is something that's sampled in nature, could this be one of those conformations that stops the aggregation? So what we looked at was the effect of TAPO1, which is this antibody, in the disease model, and actually looks as if it protects mice, and we will talk about that a little bit later. It looks as if it protects mice against conformations that are a little bit detrimental. So when we're introducing this, we say that the peptide itself seems to aggregate and form these soluble peptide conformations that drop out of solution. But as soon as it binds into this particular conformation, our hypothesis was that this would trap the N-terminal in such a position that it would not aggregate. So how did we kind of tease that out? remember when we started, we said that this TAPA1 was a weak binder, and actually it was 90 nanomolar, and we wanted something that was much tighter binding. And the second thing that we noticed is when it was binding, it was coming off quite quickly. And so we were wondering whether we could do something with this conformation to force the conformation so that that conformation was favored. And so what we did was we took the position 1 here, position 3, up to position 12, and then we designed an amino acid that would sit here. So we put in a cysteine in this position and a cysteine in that position, and we tested it out. And lo and behold, a peptide with cysteine in that position and cysteine in that position had tighter binding to TAPA1. That was interesting in
So
itself.
the goal was really keep this part together because you knew
Keep
that
this
it
part together.
liked
Yes.
kind of folding up into this hairpin type of shape. Where
Exactly.
it then folds in on itself, and I'm sure there's some H bonds and other interactions kind of keeping it together. But by having the cysteine there, then you were able to get that covalent bond that would really tie it together into this kind of like lock
Exactly.
position to really make sure it stays there.
And putting a cysteine was inexpensive because we knew we didn't need to design any chemistry. It was just a matter of putting a cysteine as part of the amino acid sequence. But we also know that cysteines reduce and then they oxidize. So unfortunately, it meant that our cysteines were not the best of bonds. And that particular structure, although it was tighter binding, something like 20 to 30 nanomolar against TAPA1, it was actually coming out and breaking up after a little while as it was oxidized and reduced. And so we decided to migrate from a cysteine to a chemical bond that would be permanently formed between these two. So we introduced a thiolinker, and that's the story of the next topic. But for now, this was the binding position, and I just wanted us to highlight some of the things that you can now do in Nanome, because in Nanome now we can do a lot of other interesting things. So, for instance, in Nanome, we can visualize our antibodies so that anybody who is looking at antibodies can immediately identify where the paratopes or the CDR sequences are. And what I'm going to do is I'm going to look at our antibody plugin in Nanome here, and I'll highlight what I want to show. So I'm just going to highlight all this, and then I'm going to run my antibody plugin. And the antibody plugin gives us this activated. And what it does, which we've now added onto Nanome, is it allows you to choose different types of annotations. At LifeArc, we used to use either Kabat or IMGT. So I'm going to select IMGT here, but you could select Kabat. So I'm going to select that, and I'll just run our antibody plugin. So it's updating the structure, and immediately what happens with our antibody structure is you can see here that it has identified already where our peptide is sitting, and around the peptide it has annotated the CD of interest to people who are interested in antibodies is the CDR heavy chain 3, where most of the interactions happen between the antigen and the epitope. So this is usually participating in most of the binding. Here it's highlighted in purple, and you can see there it's highlighted in purple.
So yeah, you were saying the purple CDR loop. Yeah, basically our antibody plugin color coded all of our CDRs here.
Yes, and effectively what it's doing is it's predicting the paratope based on a numbering system that is normally used by antibody drug discovery groups. In this case, it's using IMGT, and our plugin can also use Kabat, it can also use Choctia, and these are all annotation systems. And because at LifeArc we used to use IMGT, I'm going to highlight in MGT, and you can see here what it has done, which is very good for us, is it's highlighted the heavy chain. So if I select that, it will select the heavy chain. And in the heavy chain, it's also highlighted the CDRs from the heavy chain, which are color-coded, green, blue, and yellow, as you can see. So the important thing here is it means now when I'm looking at this antibody, I immediately understand that this is a Fab. It's got a heavy chain and a light chain. It's got the paratopes that are responsible for binding my antigen. Automatically, without having to do a lot of work, I could then use this information for me to do a little bit further design of either my epitope, which we're going to talk about right now, or I could actually design better interactions between the CDR3 if I wanted to preserve those interactions with my epitope, or other CDR chains. So as you can see here, CDR heavy chain 2 has got this tryptophan here, which sits underneath the antibody. Arguably, that is not making a big interaction, but however, there's hydrophobic interaction that might be had there. And as you can see here, this phenylalanine number 4 is actually buried into the antibody, so make it a little bit bigger, so you can all appreciate what I'm talking about.
Yep. So
as I was saying,
phenylalanine number 4
phenylalanine
right
here. It's bound in a hydrophobic pocket here, where it stacks against this tryptophan, and actually stacks pi stacking against this histidine number 6 here. So it's forming intramolecular interactions between its chains in that in the peptide, but also forming hydrophobic interactions. Here you've got this loose isoleucine here, got the tryptophan here, which are all hydrophobic pockets. You've got the proline that sits here, all driving this hydrophobic pocket for that phenylalanine. And so trapping this antibody in this conformation without breaking this apart means that you can have these favorable interactions. Now, to make it so much easier for us to look at these interactions, I'm just going to put a high quality surface on our Fab or the antibody that we're looking at. Okay, I'm just going to activate that. And let's see if we can generate some high quality surfaces here so we can see exactly where these interactions that I'm talking about are.
So question, Chido. Why in this case would you prefer the high quality surface versus the regular surfaces that also have the 2 hydrophobic color schemes?
The reason why I'm going to use the high quality surface in this case is I want to, number one, preserve some of the coloring schemes from, I want to be able to track the coloring by CDRs rather than by the normal hydrophobic things, because we need to be able to track where our epitope is relative to the paratopes. So I'm going to use that as a way of running through my structure so you can see exactly where, because with antibodies, really, the most important thing is to understand where the paratopes are and then understand the interactions. Whereas in small molecules, I will be very interested in just finding out that that's hydrophobic. But in this case, we want to see where we are. So let me see if I could generate surfaces, not for the antibody here, but just for the paratopes, or at least the heavy chains and light chain. We've generated the structure, the surface, so high quality surfaces, and we're looking here at heavy chain in green, in darker green, the light chain in the light green, and our ligand with chemical interactions that we've generated in Nanome, showing the intramolecular hydrogen bonds between the chain itself. So you can see how this is being preserved, this conformation. You can see the ionic bond here between the arginine and the aspartate number 7. You can see that this is actually a good interaction that is maintained. And deep in our pocket, you can see intramolecular interactions between phenylalanine number 4 and histidine number 6. And this pi interaction that is preserved here is key to how this molecule folds on itself. But alongside with that, you can see that histidine itself forms a hydrogen bond with the protein, and the hydrogen bonds that are being formed between the backbone and the protein down here. And so you can see this peptide has been caught in a conformation which is favorable for binding to TAPA1.
Let's just kind of zoom in, get a little bit of a closer look there. So just it's kind of like, so we have that ionic bonding you were saying with the arginine, that looks great. And then a little bit of this, really showing that really great pi stacking in there. So in terms of designing something new, I guess the important parts that you were looking at was, how do we keep this hairpin configuration, right? How do we kind of, get the structure pretty set? How do we conserve this ionic bonding, and how do we conserve this pi stacking in there? And then try to conserve as many of the hydrogen bonds maybe as was reasonable, right?
That's exactly right. So as I said before, the most interesting thing is we found the conformation. Could we trap the conformation? Could we make it always possible that it's sampling that conformation? So one of the key positions that we identified was actually this hydrogen bond here. And so what we did was we took this residue, and instead of having a pyroglutamate here, we put a cysteine and a cysteine, and they bound. And that cysteine mutant, if you want to call it dicysteine mutant, showed better binding to TAPA1. But because we knew that the cysteine was oxidized and doesn't always form a permanent bond, we then went on to talk to the chemist about what we could do. So we designed a thiolinker, the structure of which we are going to look at straight after this. Any questions?
Great. Yeah. Let's hop right in. I'd love to see that.
Fantastic. So now we've identified our conformation. We know that we can get better affinity. We wanted to find something that was more stable as a peptide that we could use to study the system more. So let's see if we can highlight. I'm going to remove this structure here, so we don't need it anymore. And what I'm going to do is going to pull up the structure of the cyclase peptide that we designed, and you will see as you go right now the structure of the cyclase peptide. And again, what we've done in Nanome is we've highlighted our CDRs. As you can see, they are notated with the CDR3 in purple here, heavy chain. This is a light chain CDR3. Those interactions that we talked about are still preserved. And if we want to run the chemical interactions so that we could just see the interactions in the structure, go here and see if we could generate.
We've got to select a few more as it's recognizing each part as a little bit different. That looks great with that cysteine there. Really kind of holds everything together.
Oh, this is the engineering.
Yes, now this is the engineered. Yeah. So instead of having the cysteine here, you can see what we did was we had a sulfur, carbon to a sulfur. So this is we call this the thiolinker to mimic the cysteine. But now we're starting in this position. And if we want to highlight, let's just label those positions on the structure. After we identified that beta 10 in our cyclase peptide, we then designed a peptide that was constrained with this non-natural amino acid here. As you can see, the position that we've put it in almost accentuates this hydrogen bond in the wild type. So what we have here is an overlay of the wild type peptide, which is with a pyroglutamate here, and our new non-natural amino acid here that forms a constraint so that this conformation is promoted. And so what happened with this peptide is it showed much better binding to TAPA1. In fact, we had a magnitude of improvement in terms of the binding affinity from 90 to about 9 nanomolar. And other assays were showing picomolar binding for this particular peptide. And so we had found an interesting conformation for beta amyloid peptide. We knew that it was binding to an antibody that had been generated against the wild type. And so we were thinking, oh, what else can we do with this? We knew that TAPA1 was really not binding to the wild type at the best conformation. So what we could do is maybe use this here, the new cyclized peptide, as a vaccine to generate antibodies, not to purify them, but to generate them within the mouse itself and see whether that was protective of the ALS or not. And so what we did was we took this peptide, we used a carrier protein called KLX, which a viral protein, which we used to carry this cyclized peptide with the linker between the cyclized peptide in this position to the carrier protein. We used this as a vaccine. And what we identified was interesting. First, we found that the bloods from that mouse could actually bind to the wild type peptide. So it means that the mouse was reacting and it was actually becoming immune to the wild type while it was vaccinated by a specific conformation that's trapped on the wild type. That was the first thing that we identified. This was very exciting. The next thing that the team did was looked for a mouse model for ALS. So as I said in the beginning, one of our collaborators was in Germany and he had a mouse model. And what we did was then vaccinate that mouse model and then track what would happen with, number one, mice that had not been vaccinated, mice that had been injected with TAPA1, and mouse that had been vaccinated with the cyclic peptide. And what was really exciting, and that's what the big story in the paper was, the mice that had been vaccinated with the cyclic peptide showed the same protection against ALS as mice that had been injected with TAPA1. In fact, it was better because TAPA1 was lower in affinity than the mice. So this vaccine cured, in inverted commas, the mice from ALS. And so this is the first time that we had identified a vaccine for ALS using the beta amyloid peptide. So it was an exciting project to be involved in.
That's awesome. So you're able to induce an immune response by having this peptide in the orange injected in the mice, and then that was able to have them generate their own antibodies that would then try to take it away and really work with this. But that showed that it actually had really great binding as well with this original white ligand. Yeah, that's amazing.
That's brilliant work, and it was actually structure driven, because if we didn't know the conformation of this wild type against this peptide, we would never have been able to design those peptides, because people have been doing it for a long time. They've been trying to figure out whether they could vaccinate with beta amyloid peptides or they could, and it was not working. But what was critical was finding out the structure of the wild type peptide bound in TAPA1, and then making this constraint here so that whatever peptide that we were vaccinating was generating peptides that epitope would be preserved. The cyclic peptide epitope will be preserved, which means somehow by holding the peptide in that conformation, it's pulling away the peptide from the aggregation propensity and encouraging protection against ALS.
Okay.
That's awesome. And did you also look at maybe some macrocycles or other sort of approaches here as well?
Yeah, I mean, that was quite seductive to start thinking, well, what if we had a microcycle? But the problem was, if you had a microcycle, you could design things that bound to TAPA1, but they were not really inducing any immunity in the mice. Because what you want in the mice is something that is as close to the natural peptide as possible, but has got that conformation, which is normally seen in a log, is not represented high enough in concentration for these antibodies to be generated against it. So
So by keeping it in the peptide, you're able to induce that immune response. And it sounds like there's some data indicating that this might be good for humans. Hopefully someday. Obviously, it's going to take time to really prove that it's safe and effective. But I'm personally very interested because I don't want to end up with having neurodegenerative diseases, and if getting something like this at some point in my life could help prevent that or reduce the amount of I guess neurodegeneration that would happen in that sort of disease. This sounds amazing.
Steve, you said you said the right thing there, right? That actually maybe there's some encouragement for human treatment. The interesting thing about this whole exercise was we were not using mice peptides, we're using human peptides. So all of this work has been done with human peptides. So what we can do now is just to hope that the same immunity that the mice is generating is also seen in higher mammals, primates, and then possibly humans.
Fantastic. Yeah. Cool. Well, any other closing things you'd like to say about our structures here before we say goodbye to our audience?
Well, I would like to just say, without the structure, as I said before, it would have almost been impossible to get here. And so understanding structures and understanding how they operate is probably the driver behind this particular exercise. We already had an antibody that was reasonable binding, but without the structure, there was no way of moving it from the antibody to potential treatment. So
we need structural biologists like Martina and your colleagues at LifeArc to help with the structure determination, and then really understanding what's going on with the structures, so that then people like you and your team could get to work and say, all right, now that we have the structures, what do we do to improve it? Right, the style linker, other aspects of it. What do we do next?
Exactly. And then, the big jump was getting that structure and understanding that it's actually a different conformation. And that drove a lot of the ideas, even at the end when we did the vaccination, people were going, we could design all sorts of other things. But if we didn't have the structure, there was no way we would have been able to get there. And actually, more interesting is a lot of people have worked on the full length of the beta amyloid peptide. And they never really got as much of affinity or even vaccination potential as we've done, only because we had the advantage of seeing the structure.
Well, this has been an awesome deep dive. I've certainly learned a lot about how these antibodies bind with the peptides, how creating new peptides could help lead to a better immune response that could hopefully prevent big neurodegeneration diseases that are caused by these misfolded peptides that sort of aggregate together. But yeah, this is really amazing. I look forward to seeing this progress, hopefully into the clinic, and help out real patient lives someday.
Yes, thank you very much.
Thank you, everyone.
Thanks for tuning in to share. Fantastic. Thank you.
Thanks, everyone, and we'll see you in the Science Metaverse.
Ciao, ciao.